Serological detection techniques of plant viruses. So now you will see what are all the different types of serological techniques are employed on the detection and identification of plant viruses. My name is N.H. Shankar Reddy and I am doing PhD plant pathology in Annamalai University. So here, serology is a branch of science that deals with the immunology or antigen antibody reactions. So due to this antigen antibody reactions that we can easily identify the particles of plant viruses or the depositions of plant viruses in the given sample. So this technique or this serology can be widely used in agriculture, horticulture and forestry and immunology techniques. These are extensively used for the detection of different types of plant pathogens, especially plant viruses. So now we'll start with the serology definition. So based on the antigen antibody reaction, this serology actually works. Here, antigen antibody react. There are let them react on the particular given sample. Let us consider if the particular sample has containing of virus. Now we are giving the antigen from the outside so that it will react with the antibodies of plant viruses, so that it will so it's easy to identify the samples. So here, due to this antigen antibody reaction, there is a bridge is formed here. Let us consider if it is a antigen, so the antigen which are giving from the outside can be detected by the plant antibodies, so that a bridge is formed or the connection or link is formed between the plant pathogens and as well as given that we are giving the antigens from the outside. So due to that, a reaction can be formed and we can identify the presence of plant pathogens, that is especially plant viruses. Here, before entering into what are all the different serological techniques employed in plant virology, now we will see some basic definitions, how it actually works and what are all the things. So the first one, we will start with the antigen. What is antigen? So actually in in electron microscope in the earlier the production of antigen itself, and now we discussed what is antigen. So yeah, I am coming again. So here the antigen is a substance that evokes the production of antibodies. So it is actually a foreign substance that we are injecting from the outside that evokes or that initiate the immune responses, that is antigen. So maybe this antigen may be proteins or maybe polysaccharides or maybe lipids or carbohydrates or nucleic acids or enzymes or toxins, whatever it may be, that we are injecting from the outside, which act as an antigen or that evokes the production of antibodies from the inside. So each antigen is made up of distinct subregions. Let us consider this is antibody. Each antigen consists of a distinct subregion, like shapes like this, maybe like this, or different distinct subregions will be there that have a definite spatial and electronic configurations. So if it has this type of shape, it can only bind to this type of antibody structure present on the antibody. So if the structure will be like this, that then only the antibody can find the antigen, like that. That's their telling their distinctive subregions are for the specific electronic configuration, so that it can be bind with the specific antibodies. Here these some restricted regions of the antigens that simulates the antigenic responses and are the regions in which antibodies are get attached. That's their telling. Antigen stimulates the antigenic responses. There are restricted regions. Let us consider if the particular antigen that we are injecting outside forms a globule like this. So antigen has to produce like this. Simply a enzyme and key hypothesis. And simply enzyme and key. If this type of structure can be produced by the antibody, so that can bind with the specific antibody or specific epitope. So this is about antigen. Note an epitope. Here they are telling these regions are called antigenic determinants or epitopes. This particular binding sites are, this is called epitope. What they are telling is epitope is also known as antigenic determinant. This epitope can determine the antigen. So it is a part of antigen that recognized or specially by antibodies. That's why I'm telling, if they are producing this type of structures, the antibody will produce like this, so that it can bind the particular epitope regions. If they are producing globule like this, the antibody has to produce structures like this that they can trap or that can they bind. So this they are telling is particular epitope regions. This region is called particular epitope region. The The second one is antibody. So antibody actually which are produced by the B lymphocytes that are present inside our body. So it is actually a Y-shaped structure or molecule consists of a 4-peptide polypeptide chains. We can see here. This is a polypeptide chain 1, 2, 3, 4. We can see here this is the specific epitope binding regions. If they are producing this type of structure, the antibody has to become like this so that that can trap this type of antibody. So they are producing different types of structure we can see here. So according to the shape, according to their epitope region, the antibody has changed and can trap the exact antigens here. So actually this antibody is a Y-shaped molecule. It consists of 4 polypeptide chains. This is a chain 1, chain 2, chain 3, chain 4. So these are also called as antigen binding sites. These are also called as antigen binding sites because antigen come in contact with this and antigen binds on the specific sites with the specific epitope region. So that is called, these 4 polypeptide chains are called as antigen binding sites. So the specific protein formed by the warm-blooded animals, especially humans. We are warm-blooded animals, yes. So it is formed by this warm-blooded animal in response to the specific injection of antigen. So why we are injecting antigen? In some cases of the research or some cases that we are injecting the mild strains as like of a cross production or mild strains or some some antigen, so that can evokes the responses of immunity. So that whenever the pathogen come in contact and the next time the immunity responses will immediately activate and it can kill that particular antigen, so that disease can be eliminated, or maybe some infections or whatever, it can be eliminated. So response for the specific recognition site and elimination of the antigen. This is the specific recognition sites and shapes. So this antigenic sites or polypeptide chains can respond to the specific region or the specific epitope or the specific shape of the antigen, so that the antigen can be eliminated. So antigen can't able to produce its action, so because of the antibodies production. This is about antigen and antibody reactions. So wherever we go, whatever the protein based technology, mostly this antigen antibody reactions will be only there for the detection and as well as identification of plant viruses. So these are the different types of immunoglobulins are used in the plant virology, that is IgA, IgD, IgE, IgG and IgM. These are the different types of immunoglobulins which are used in plant virology. These are all the shapes of antibody complexes, how they will be like that. So here in virology, most widely used immunoglobulin IgG, very important. 70% of the medical immunoglobulins are the required immunoglobulins are produced are the shape of IG, so with the class of IgG. So most of the immunoglobulins are belong to the class of IgG. Nearly 70 to 80% of the immunoglobulins that are comes under IgG, very important. So the types of antigen and antibody reactions. Here there are two broad categories, are the two broad senses of antigen antibody reactions are there: homologous and heterologous. Homologous in the sense the reaction between anti-serum or antigen that is caused by the production and resemblance of fit, lock and key hypothesis. Let us consider if the antigen shape is like this, the antibody will also produce the same shape like this, so that it can fit to the antigen, so this is that they are telling homologous reaction between the same type or production of antigen and antibody reactions are that can fit like a lock and key hypothesis, exactly fit to the lock, lock and key exactly fit to the lock. That is considered if the antigen is a lock, sorry, if antigen is a key, the antibody is a lock, it is produce the same shape or structure will be there, so it can lock the antigen, sorry. Here in heterologous reactions, the reaction between antigen and antibody closely resembling but not identical to the one that causing the production of antibodies. Let us consider if it is producing the shape like this, it is almost closely resemblance but it not identical, it is not exactly identical to the one that is responsible for the production of antibodies. So this is the two types of reactions, homologous and heterologous. Homo means same, that is a reaction between the lock and key hypothesis, exactly fit to the antigen that is coming from the outside. Whereas in heterologous reactions, it is almost resembling but it not identical to the exact one that is responsible for the production of antibodies. So here coming to the serological techniques, There, as I told you earlier, it's mostly antigen and antibody reaction techniques. There's several other ways also used for the detection of antigenic substances that can carry them. So here, protein-based techniques are especially employed for detection of plant viruses, especially that can detect the virus code protein. So this technique can be used to detect the virus code protein. Coming to in composition of plant viruses, virus composites are two major compositions or two major structural outlets. That is, first one is nucleic acid, second one is protein coat. Here the protein coat. So this protein based techniques can identify the protein coat or coat protein. So here this phases of reactions are two types, that is liquid phases and solid phases. In liquid phase reactions, precipitation test, agglutination test, ortolone double diffusion test. These are all different types of liquid phases that comes under serology. Coming to the solid phases, enzyme linked immunosorbent assay, sodium didosyl sulfate polyacrylamide gel electrophoresis, otherwise called as SDS phase. So it is a variant of phase normally used for the detection of proteins, and I will tell you how it will work. And immunosorbent electron microscopy, Western blot, and dot immunobinding assay and tissue immunobinding assay. These are all the protein based detection techniques that comes under solid phase. Now we will see one by one. In liquid phase, the first one is precipitation test. The name itself indicate when we mix antigen and antibodies, they combine and they form precipitation, or the precipitation reactions will happen. So this precipitation test can be widely used in detection of plant viruses in plant virology. So how this precipitation work now we will see. When we come in contact, or when we add two that is antigen and antibody, so the reaction will happen. What different types of factors are involved in between these reactions. So the main factors are dependent in this precipitation reaction is the salt concentration, how much amount of salt concentration will be there, the pH, temperature, and the presence of interfering compounds, and the ratio and concentration of antigen and antibody reactions in this. These are all the factors that can influence the precipitation test. So when we add antigen and antibody, different types of frothing substances or precipitation substances formed. So based on the different types of factors, as I told you earlier, I mentioned you. So by using this, we can identify the plant viruses. This is one of the liquid phase tests, that is precipitation test. The second liquid phase technique is ortolone double diffusion technique. Actually, in this technique, what we are doing, the plant extract or plant sap initially has to prepare. So how the plant sap has to prepare in sap preparation, or the preparation of sap material, I already explained. So that we have to grind it by using macerating jar, by using macerating jar or some other techniques. So we can make it into plant sap, and we can by using a muslin cloth we can have the extract. So initially we have to get the extract of it, and then in double diffusion technique, high quality grade agar can be used of 0.9%. Actually, now we are agriculture or bacteriological agar that we are using is concentration of 0.8%. 0.8%. So here the high grade agar can be used, that is 0.9% of agarose, consists of 0.9% of agarose, or iron agar number 2 can be used. This is a high quality agar, and it is prepared by the buffer which is a pH of 7.6, and also it is containing a 0.8% sodium chloride which act as a preservative, which act as a preparative buffer, and preservative also can be added to this, that is 0.02 chemical of sodium azide. So I'm not going in detail, but I'm just giving the outline so that you will identify what all the techniques are there in the identification of plant virus. I'm just touching on just like an outline, so that will be easy. So what are the techniques will be are there to identify. First of all, if you know the what are all the techniques will be there, it will be easy to go in depth. So now you'll see here during this ortolone double diffusion technique. So plant extract, or the whatever the sap extract that we are using, are placed at inside, placed at the center. So different antisera which are let us consider, which consider different types of which different types of holes, or can be some arrangement will be like this, that we are placing the sample at the middle. So on the remaining anti-sera that we are placing here, anti-sera that we are already prepared, so the anti-sera that we are placing here. So if you place the anti-sera here, The various patterns of precipitation lines can be obtained. This precipitation lines can be obtained based on the relationship between the antigen, or maybe some elongated virus may be diffused rather poorly, or some viruses may be diffuse very good with this particular anti-sera, and so that we can identify different different precipitation regions, as well as different patterns can be identified. So this technique can be used to modified either at a low concentration of agar, that is 0.5 percentage, at this low concentration, that is 0.5% concentration also can be used this technique. This is orchidal double diffusion technique. And the third one is chloroplast agglutination test. See, in this chloroplast agglutination test, the test can be performed on the crude extract of a suspected virus leaf or plants. Initially, as like of earlier experiments, so we have to initially get the infected plant sap. So infected plant sap or extract initially we are preparing here, but as I told you earlier, macerating and going through some muslin cloth, and we'll get some juice along with the infected plant viruses. So the plant material grounded to produce a sap suspension of chloroplast and the cell debris. It also contains chloroplast as well as cell debris. Now anti-serum is added or suspended into the virus particles, or that is a sap containing the viruses. So let it allow for some times, or maybe 5 minutes or 10 minutes, maybe time varies dependent on the reaction. So if virus is present, cloudy chloroplast agglutination is visible like this here. If the virus is present, cloudy chloroplast agglutination can be seen here. If no virus can be present, it won't react. We can see here. If no virus, no reaction. If the virus is present, a cloudy chloroplast agglutination can be visible. This can be matched with the presence of virus. So here we are initially preparing the sap, and anti-serum is adding to the sap, because the sap also contains of viruses as well as chloroplast material, so that it can react with that particular virus, and we can come to know that the presence of virus. This is chloroplast agglutination test, comes under liquid phase. So the final one is latex agglutination test. So in the name itself indicates, so the anti-serum is absorbed onto the polystyrene latex beads. So latex beads will be, we can see here latex beads, these are latex beads. So anti-serum actually is absorbed onto the coating. You can see here anti-serum absorbed on the top of the latex beads. So here it is mixed with the crude sap. So the sap has to prepared as like of other techniques. So after preparing the crude sap, so this latex bead has to suspended with the virus infected materials, or the some materials that we feel, or that we intended that the sample contains a plant virus or something. So that we are suspending the and this anti-serum coated latex beads. So let it allow for sometimes, or this consists of a round latex beads where the antigen or where the anti-serum is attached here on the top of the latex beads. So the flocculation can be happens here because of the antigen antibody combinations. Here this allows the detection of 100 to 1000 fold smaller quantity of plant virus. Even the very small quantities of plant virus can be identified by using this latex agglutination test, and moreover it takes very less time, that is 5 to 10 minutes. Within 5 to 10 minutes we can identify this presence of virus. So this technique is the most sensitive technique of the precipitation test that we have seen earlier. Among the all precipitation techniques, this latex agglutination test is a very sensitive precipitation test that we are using in plant virology. So here when the antigen concentration is low, precipitation does not become visible. In such cases, indirect tests are performed to find out the combination between antigen and antibody. In some cases, at a very low precipitation, we can't able to visible the plant viruses. At that time, what we are doing, so to find out the reaction between antigen and antibody, some of the indirect tests will be there. So we have to perform that indirect test for the identification of plant viruses. So now coming to the second phase, that is solid phase. So what are all the techniques that comes under solid phase? First one is ELISA. ELISA in the sense enzyme linked immunosorbent assay. This is one of the technique which is based on antigen antibody reactions, which is used to identify the plant viruses. So precipitating or agglutination is required, or maybe enough amount of antibodies are required. We can see this in precipitation test or agglutination test. Enough amount of antibodies of plant viruses is required to be present to identify the plant viruses. But in ELISA, very small quantities or very small concentration of plant viruses is antibodies is enough to detect the plant viruses. That is, it is called, it is one of the most sensitive technique for the detection of plant viruses. If you see the sensitivity, it is just 1 to 10 nanogram per ml of liquid or sample. This is the most sensitive technique that we are using in plant virology for the detection of plant viruses. So in this question that I had seen this question many times in many exams, especially national eligibility tests. So what is the most sensitive technique used for the detection of plant viruses? That is ELISA, not PCR. If you take the PCR, the sensitivity of PCR is 1 to 10 picogram per ml. Picogram is bigger than nanogram, that we have to consider, or we have to know about this, the dimensions, picogram, nanogram, milligram, all these things that we have to come to know. Then only we can identify which one is big and which one is smaller. So the sensitivity of ELISA is 1 to 10 nanogram per ml, whereas the sensitivity of PCR is 1 to 10 picogram per ml. So different types of ELISA will be there: direct ELISA, indirect ELISA, sandwich ELISA, competitive ELISA. Most of this work on, most of this relies on antigen antibody reaction only. So this technique was initially discovered by Clark and Adam in 1977. So this plate consists of a plate like this. It consists of small holes. Nearly 96 holes are present. That is 12 into 8. 12 12 12 12 12 8 will be there. That is 12 into 8 equal to 96. Total 96 wells are present. This is the basic standard plate that we are using in ELISA. The second one is, now you'll see what are the advantages, some of the advantages of ELISA. It is a very simple technique and it's very easy to perform, and it's a very fast method when compared to other techniques. Compared to other techniques, it's a very fast method. So the sensitivity, as I told you, is 1,000 times more sensitive than other any other serological and other techniques that are employed in plant virology. Not only in plant virology, that is employed in almost on all biology. If you take any technique that is not sensitive than ELISA, because it is 1,000 times more sensitive than the any other techniques in the detection of plant viruses, or than any other conventional electron microscopy methods. So it will take very less time for the detection of plant viruses. That is just within 2 to 3 hours that we can perform a large amount of samples. So commercial kits also available in laboratories, or on out, if you go, the commercial availability will be very it will be easy to get the ELISA kit. So very small volumes of extract may be preferred. Even though very small amount of volume, that is 0.1 microliters, is sufficient for this performing the ELISA technique. So the second solid phase is SDS phase, sodium diazoxide sulfate polyacrylamide gel electrophoresis. It is a type of phase, that is polyacrylamide gel electrophoresis. That is SDS phase is a type of page technique. So the main aim of this SDS phase is separation of proteins based on the size and shape. It's a very important. The main aim of this SDS phase is separation of proteins based on the size and shape, isolation, purification of proteins required by the study of various functions of the organism. If you purify and isolate this protein, that will be helpful for the study of the various functions of the plant pathogenic viruses. So when proteins are separated by using this electrophoresis techniques or gel matrix, here the important thing is small amount of proteins migrates faster because of their small, let us consider, if you take two men which is weighting, one man weighs 50 kilos, the second man weighs 150 kilos. If you raise them to, the 50 kilos man will run very faster because of their body size and bulk and density. Mainly I can tell as a density. Here that the same thing will apply. Small proteins migrates faster due to less resistance to the gel matrix, whereas bigger proteins will move very slowly. In SDS phase, the use of sodium diazoxide sulfate, that is SDS, is also known as sodium lauryl sulfate. This sodium diazoxide sulfate is also known as sodium lauryl sulfate. Is a chemical that is used in polyacrylamide gel electrophoresis. So this can eliminate the influence of the structure and large charge and proteins are separated solely by means of polypeptide chains length. So how the polypeptide chain, whether it's a big or small, maybe how it's a shape or size, it can be varies from protein to protein. So by depending on this technique or by taking it as an advantage, this sodium diazosyl sulfate will work and identify the different types of plant viruses. So we can see on the right side, we are injecting the sample into the wells of gel. Then based on the protein size, it will run. So small protein migrates faster than the big protein. Third one is immunosorbent electron microscopy, that is ISEM. Immunosorbent electron microscopy is first discovered by 1973 by Derrick. So he published in a serological specific electron microscopy method. Later it was simplified, or now we are using it as immunosorbent electron microscopy. The main principle of immunosorbent electron microscopy is selective trapping of plant viruses and electron microscope grids. I know in electron microscope, in the production of anti-sera itself, now I told you that in here the viruses are actually absorbed onto the biofilm coated nickel or copper grids. So here the selective trapping of the viruses on the nickel or copper grids, this is the principle mainly involved in the immunosorbent electron microscopy. So this is a highly reliable and very fast and sensitive as like of viruses for the detection of as like of ELISA for the detection of plant virus. It is also one of the most sensitive technique as like of ELISA for the detection of plant virus. Fourth one is Western blot. In Western blot, usually used for the characterization of plant proteins, that is virus proteins. This blotting technique or Western blotting technique is employed for the identification of proteins. So it is an advantages of over advantage of determining the plant proteins, that is viral proteins and serological or molecular properties. So if you identify these proteins, that is easy to study the molecular properties and as well as protein structure of plant viruses. Actually in this method, so actually the antigen or the protein antigen is transformed into polyacrylamide gels, a special type of gel where it was instead of following the electroporosis or nitrocellular membrane or nylon membrane, we are doing it on a polyacrylamide gel. That is a special type of gel that we can transfer into the protein, and you can perform the electroporosis technique as like of how we are giving the electrical charge and the on the density proteins will move faster, like that we can identify. The fifth one is dot immunobinding assay, that is Tebow. So it is require very simple and easy method for the detection and identification of plant viruses. So we can apply this on a samples are applied to the nitrocellulose or nylon membranes. The samples are initially applied to, let us consider it is a type of nitrocellulose or nylon membrane. The samples are applied to nitrocellulose or nylon membranes. So antigens also can be added to this. So the samples containing the virus antigens are prepared by the grinding tissues. Initially the tissues can be grinded, that is the lab samples or leaf samples can be grinded. So this contains an antigen, the contains virus antigens are prepared by grinding the tissues, and tris buffers and saline, all things can be applied by using antigen and antibody reactions. The pathogens or the presence of virus can be detected. This is about dot immunoblinding assay. And the last one is Tebow, that is tissue immunobinding assay. It is used for the detection of virus antigens. So the plant extracts such as leaves, stem, bulbs, tubers, roots or fruits or insect vectors of plant viruses can be presence of this can be identified by using tissue immunobinding assay. So this is about different techniques, or what are all the different types of serological techniques that are employing in the detection of plant viruses. I'm not detailed, I had given the detail and in-depth description about this. I just want to conclude what are the different types of techniques are there for the detection and identification of plant viruses. This is a just outline, so that you will have an idea how what are all the techniques that are employed in serology for the detection of plant viruses.